ওখানে বলছে ওয়েটিং ফর আশুতোষ কলেজ অফিসিয়াল, মানে ওই টাইমটা দেয়া আছে, এখন অব্দি স্টার্টিং দেয়া নেই, 9টা স্টার্ট করেনি।
9টা, হ্যাঁ 9টা বাজে, তো তাহলে একবার পোস্টঅফকে জিজ্ঞেস করেন। পোস্টঅফ শোনা যাচ্ছে?
স্ট্রিমিং স্টার্ট হয়ে গেছে।
Okay, fine. Thank you so much. That is all I wanted to know. Thank you. Let us begin today. A very good morning to all of you at the Host Institute, and a very good evening to Abhisek, who is joining us from the US. It gives me a great joy to share with you that we had embarked on this journey of AAHAN in way back rather in April 2021, and we've already reached the 3rd chapter today. And I take this opportunity to welcome our speaker for today, Dr. Abhisek Bhattacharya. On behalf of the Postgraduate Department of Zoology, Asutosh College. It gives me immense pleasure to introduce him today, who is working right now as a postdoctoral fellow at NCI NIH USA, and actually takes me down the memory lane when I first met him as a very aspiring and a budding researcher, and who had just begun his quest to study the basic sciences after completing his MBBS degree. He has had a very illustrious academic career, which not only includes an MBBS degree with distinction from Calcutta National Medical College way back in 2006, but after that, he followed it up with a master's in genetics from the University of Calcutta in 2008, where he also stood first as first. He did his Ph.D. in immunology from Baylor College of Medicine, Houston, Texas, USA. He's currently pursuing, as I've already said earlier, his postdoctoral research, and is also a recipient of several honors, scholarships, and recognitions for his scientific contributions and his academic excellence. Actually, we share a very common interest, that of genetics, and it is so rewarding to see him today that he's working at this position right now. Congratulations once again, Abhisek. Now, he's also made several publications in very reputed journals for his doctoral dissertation, which happens to be the role of autophagy in the innate immune cells in mediating inflammation and autoimmunity. He's currently working on the molecular basis of MHC restriction of the alpha-beta T cells, and is also a reviewer in esteemed journals such as the Journal of Genetics, Biomolecules, Immunology, Pharmaceuticals, to name a few. Apart from being an avid researcher, he is also having a keen interest in teaching and has been taking regular classes at molecular biology, immunology, and gene regulation at the Baylor College of Medicine. Now, I can go on speaking about him, showcasing his multidimensional persona, but I suppose due to the constraint of time across the globe, because we are only kind of starting our day here, it's almost nearing midnight at his place. So I suppose we should now focus on his lecture today. And without any further delay, I'll ask Abhisek to go ahead with this presentation. And the discussion that he will take up today is basically the autophagy in the innate immune system. Now, we have basically divided our pool of students into 2 batches. The postgraduate students of the SEM 1 and the SEM 3 are with us on this GMeet platform. And our undergraduate students who are from SEM 1, SEM 3, and SEM 5, of all of them are basically joining us in the YouTube live streaming right now. So they are also part of this presentation. So I hope my students can get inspired by your work, and also they can learn a lot from this presentation. Of course, looking forward to many more of this. So I welcome you once again, Abhisek, and you can now start your presentation. Thank you so much for joining us today.
Thank you, Didi.
Thank you.
First of all, it's a great honor. More than that, it's a privilege to be here today. And I'll start with the same thing that I said during my PhD dissertation. Where I am today can largely be attributed to Sriparna Datta. Had I not been with her, most likely I would either be an MD treating patient. I don't know if that's better or not, or I would be an actor on stage somewhere. I am here, and it's largely because of Sriparna Datta. So it's a great privilege to be here today in front of our other students to present my work. So without further ado, let's start with our thingy. Okay, here we go. I hope everybody can view the presentation. In case of any problem, just let me know, because I cannot hear or see.
No, you're absolutely fine.
Thank you.
You're absolutely fine.
Okay, so my topic today is autophagy in the innate immune system. So the immune system is divided in two basic components, the innate immune system or innate immunity and the adaptive immune system or adaptive immunity. And the basic difference between these two systems is the presence of immunologic memory. Adaptive immune cells have immunologic memory, innate immune cells don't. So anything that has a memory is an adaptive immune cell. What is this memory thing? Upon secondary exposure, adaptive immune system responds faster, stronger, and specifically to that antigen. Three things that characterize immunologic memory: faster, stronger, and specific response upon secondary exposure. And this happens because adaptive immune cells have antigen receptors, clonally distributed receptors. None of these things are present in the innate immune system. They don't have memory, though recently there is something called trained immunity, but it's completely different. Bottom line, innate immune cells don't have antigen receptor, don't have immunologic memory. And that's the whole focus of today's lecture. My postdoctoral work is mostly in Dr. Singer's lab. It's in this part, alpha beta T cells, MHC restriction, and immunologic memory. But today I'm going to present most of my thesis research in Baylor College of Medicine as a PhD student. So when we talk about innate immune system, we divide it into three basic protection components: barriers, cellular innate immunity, and humoral components. The barriers involve skin, epithelial cells, mucosal layers, and things like that. I'll very briefly touch on the epithelium in this case. The major focus of today's topic will be cellular innate immune cells, innate immune system. But there is also a big component of innate immune system, the humoral immunity, including complement and some other secretory mechanisms. We'll just touch a few of those. So what about cellular innate immune system? So basically the cells are divided into two categories: granulocytes and agranulocytes. As the name suggests, granulocytes means cells that have granules. There are three major cells in innate immune systems: neutrophils, basophils, and eosinophils. They have granules. There are also mast cells and NK cells. They also have granules. Now, when it comes to agranulocytes, the term not only comprises innate immune cells, but basically in a PBL or peripheral blood smears in human, when you stain for H&E, hematoxylin eosin staining, most of the agranulocytes are actually lymphocytes, T cells and B cells. But innate immune cells, macrophages and dendritic cells, are cells that don't have granules. They are also agranulocytes. There are very few of these in the bloodstream. So when you actually stain, this is what it looks like. The right side of the screen, my right, actually the left, the lymphocytes, very high nuclear to cytoplasmic ratio, no granules. And when you can see neutrophils, eosinophils, and basophils, they are full of granules. In neutrophils, it's not that clear because it's neutrophils. In an H&E staining, you don't see those very clearly, but you can clearly see in eosinophils, basophils, they're just full of granules. Any stimulation, the first thing these cells do is to release their granules. This is an evolutionary protection mechanism, so they don't have to synthesize the antimicrobial products. Those are already stored. You touch them, a bacteria comes, a bacterium just pinches on them, they immediately secrete them. That's the major protection mechanism. Doesn't mean they don't do other things, and we'll go to that other things as well. But this is how they look like. You have plenty of granules in there. So how do these innate immune cells actually work? It starts by sensing. There are two themes in this innate immune system: danger and stranger. Actually, it's a very famous model proposed by Polymath Singer. Stranger, as the name suggests, foreign materials. Danger are intracellular inflammation inducing products. The basic mechanism by which all innate immune cells initiate their program is by sensing the danger or strangers. Once they sense by different receptors, different mechanisms, they migrate to the site and then do their effector functions. There are some subdivisions, but more or less all innate immune cells are capable of doing some things in common. For example, all innate immune cells, neutrophils, macrophages, dendritic cells predominantly, do phagocytosis. No, other cells, not so much. Degranulation is the release of granules into the extracellular milieu. Of course, if you have granules, you will do it. Mast cells, neutrophils, eosinophils, they will do that. Macrophage or dendritic cells, they don't have granules. Cytokine secretion is common for all innate immune cells. When they're stimulated with appropriate stimulus, they will secrete cytokines. Antigen presentation, classically dendritic cells, and it will be a component of today's talk. And then there are some very specific mechanisms. I'm not going to go into details about any of those. Just to mention, one of those recently discovered is called netosis. When neutrophils recognize a bacteria, one of the strange mechanisms is that they die, but they make, it's like a network of DNA. It's very sticky. It's exactly like spider web. They trap the microbes in there. Not going to talk about that either. So we'll predominantly focus on phagocytosis, but mostly on degranulation. That's my topic, and some secretion and also antigen presentation. All these things are related intracellularly to the process called autophagy. So what exactly is autophagy? Let me introduce, this is a very hot topic right now, has been for quite some time. So what exactly is autophagy? It's an intracellular degradation mechanism. A membrane develops within the cell, a double layer membrane called isolation membrane. It engulfs multiple things, damaged organelles, bacteria, anything that's there, it will just engulf it, form a complete circular autophagosome, then it will fuse with the lysosome, and there the degradation will take place. Following degradation, they will use the product, the amino acids and things for recycling and energy. So this process was discovered as a mechanism against starvation. I'll come to that, but please remember this slide. This will be the hallmark of our presentation. I'll come back to it again and again because a lot of things, a lot of mechanisms have been or shown to be involved in multiple steps of this. So when autophagy was first discovered, this is the famous scientist Yoshinori Ohsumi in Japan, and he got the Nobel Prize in 2016 for the discovery of autophagy. They discovered autophagy in yeast cells by one of the strongest characterized stimulus, starvation. So on the left side, this is the electron microscope of a cell, a yeast cell, when you keep it within the normal growth medium. 30 minutes of starvation, you can already see the small vacuoles appear in there. These are the autophagic bodies. 3 hours, you see more and more of this. And 8 hours starvation, there are lots and lots of these products inside the cells. These are all those membrane components. They take up their intracellular products, digest, start releasing amino acids and things like that so that the cell now can respond to the starvation. So this is how autophagy was developed. But then it turned out starvation is not the only stress mechanism that will induce autophagy within the cell. Don't go into detail. This is a review where we actually showed multiple things. So the first one discovered is called nutrient sensing module. It will induce autophagy. Oxidative stress will induce autophagy. Hypoxic stress will induce autophagy. And ER stress will also induce autophagy. So now autophagy is regarded as a stress response pathway in the immune system. Multiple forms, anything, most stress response, including infection, will induce autophagy in the cells. And that's considered one of the evolutionary conserved mechanism from yeast to higher mammals. It's the same process, same pathway, everywhere it's the same. There are many, many, many molecules that are involved in induction of this pathway. I just want to focus on two different things in this slide. This is again from a review that we wrote a long time ago. Each step involves multiple mediators. ATG stands for autophagy related genes. All these products are involved in the autophagy pathway. Doesn't mean that these genes only mediate autophagy. For example, LC3 is one of the very early inducer of autophagy, but it's also involved in phagocytosis, completely independent to the autophagy pathway. It's called LAP, LC3 associated phagocytosis. So what I want you to take from this slide is that there are multiple proteins involved in autophagy pathway, but these proteins also mediate other intracellular functions. So with all of this, let's focus on two of these. Just remember, ATG5 and ATG7 are two things, two important proteins. Both of them are involved in the initiation of the autophagy pathway. You block them, you block the induction of autophagy within the cell. This is the way we used to examine autophagy in mouse models. Now, I told you I'll come back to this whole process again and again, this fusion thing. You can immediately see when this isolation membrane develops within the cell and engulfs intracellular component, it can also engulf bacteria or even viruses. So this is one of the mechanisms by which intracellular autophagy protects cell from bacteria. And also bacteria develop different mechanisms to inhibit autophagy for their survival. I'll just give you one example that we just published recently. This is in epithelial cells. This will be the only example of epithelial cell that I'll provide today. But this mechanism is not specific for only epithelial cell. Bacteria co-evolve with the host, and this is one of the very common mechanisms. So what they do, if you have this bacteria, Pseudomonas, one of the major infectious agent, particularly after hospitalization, if you have this inside the cell, the bacterium comes in, there is this isolation membrane, it engulfs the bacteria, degrades it. So the bacteria actually develops a very specific secretory component which will inject a toxin. There are many toxins from this bacteria, but a very specific one called XOS. This XOS toxin will inhibit this isolation membrane formation and development into autophagosome. So you will have the bacteria in the cell, but you cannot form this autophagosome, cannot digest. So the bacteria will survive within the cell. So this is one of the mechanisms by which most innate immune cells, especially epithelial cells or barrier cells, they can counteract bacteria development or bacteria propagation within the cell. So far, we have just the basic differences between innate adaptive immune system. I just mentioned components of innate immune system, innate immune mechanisms, and the autophagy path. Now let's go into the innate immune system in more details. So the innate immune cells sense these danger or stranger molecules by pattern recognition receptors. One of the major one is the toll-like receptors, or TLR. But TLRs are not the only ones. There are three broad groups: extracellular or intracellular, TLRs, toll-like receptors, NLR, intracellular node-like receptors, and RLRs, RIG1 or retinoic acid inducible receptors. All of these have been shown to somewhere merge or directly induce autophagosomes or autophagy pathway. TLRs was first discovered in our lab, in Tony Issa's lab in Baylor. Not the TLR, but the TLRs induce autophagy. And then NLRs and RLRs come in. So whenever an innate immune cell encounters a bacteria, this they either intracellular or extracellular, they encounter the bacteria via one of these pattern recognition receptor. They do multiple things, and one of the most important things they induce within the cell is the autophagy pathway, always, because they want to clear the bacteria. So how these innate immune cells clear the bacteria, induce autophagy, and do phagocytosis. So phagocytosis means they're taking solid products from outside the cell. Endocytosis is cell drinking liquid products. Multiple labs, including ours, showed that it's the phagocytosis pathway that intersects with autophagy pathway. Endocytosis, not so much. So again, same thing, same diagram. So you have autophagy going on inside the cells. The phagosomes comes from outside. Somewhere the autophagosome fuses with the phagosome, or the autophagy proteins directly bind to the phagosome and causes a process called phagosomal maturation. That was the part which was known when we started our project. So TLRs get induced, somehow ATG5 and ATG7 goes to the phagosome, cause phagosomal maturation, and finally degradation. But what we showed for the first time, and then it became very clear and a very common mechanism, that there are many indirect effects of autophagy on phagocytosis and other processes as well that just we started to realize right now. For example, what autophagy does, it degrades proteins or organelles. So what happens when the cells don't have autophagy? These proteins that were supposed to be degraded are now within the cell. One of those proteins is called sequestosome or P62. Turned out when you have a lot of P62 accumulation within the cell, it activates a transcription factor, NF2L2. And that transcription factor can go inside the nucleus, start transcription of phagocytosis or receptors that can mediate phagocytosis. And those receptors plays a big role in clearing different bacteria, including tuberculosis. So this was a completely new avenue where there was no direct effect of autophagy on phagocytosis, but there are indirect effects. And later on, multiple different labs in different all over the world, they showed it's not just phagocytosis. When you don't have autophagy, you have accumulation of different things within the cells. And those different things mediate different functions, including initiation or inhibition of transcription. So that's where autophagy in innate immune cells intersects with phagocytosis. So now let's focus on the main thing that I want to show, degranulation pathways. So I showed this picture, neutrophil, eosinophil, basophil. 60% of a human blood peripherals, those are neutrophils. But when we started our project, we had a huge long list. This is from another review. I just want to show some strange things. A lot of cells, these were known that autophagy inhibits secretion or degranulation from these cells. Nobody ever studies the most important one, neutrophils, because neutrophils, as I mentioned, are most common in our bloodstream. This process later on by us and other labs, we started calling it secretory autophagy, a completely different role of autophagy in mediating. So neutrophils have four basic types of granules: storage granules, then tertiary, secondary, primary. These are the product that's stored in the granule. You stimulate neutrophils, take supernatant, assay for these products, and then you see whether you get enough granular content outside the cells. So we made this autophagy deficient mice, and first one we did was the ATG7 deficiency. ATG7 deficiency, there is no initiation of autophagy, so these mice don't have autophagy pathway functional. But again, please remember ATG7 can do other things as well. So we generated our control or ATG7 knockout neutrophils. Actually, this was a myeloid-specific knockout, which means not only neutrophils, but neutrophils, macrophages, they all lack autophagy in there. So we take these neutrophils and stimulate with a bacterial tripeptide with drug. This is FMLF. This is the methionine leucine. Actually, this is phenylalanine, but always written as F. This methionine is formylated, and this is a very strong stimulus of neutrophil degranulation. This is only found in the bacteria, never in mammalian kingdom. So whenever neutrophil encounters this tripeptide motif, they realize there is some foreign materials in the body, pathogens. So they immediately degranulate. So all their granules are now outside the cells. So we collect the supernatant and measure different products. So the first one I'm going to show you is the product of tertiary granules. It's called gelatinase. This is a gelatinase zymography. So you can see without stimulation, there are very little products in the supernatant. Each lane is one mouse. So these three are the ATG7 control. So they have ATG7 gene within them. So when you stimulate, they secrete a lot of gelatinous in the supernatant. These three, written as ATG7 myeloid negative, so these cells don't have ATG7. They still secrete, but not as much as the wild type control. And when you statistically show this at different time points, 30 minutes or 15 minutes after stimulation, you can see lack of ATG7 reduced their degranulation. Now you have very little product in the supernatant. So this is for the tertiary granules. We found this exact same thing for the secondary granules. Lactoferrin is even more striking. There is very little lactoferrin secretion in the supernatant. And also the same thing for the primary granules, beta-glucuronidase. Again, 15 minutes or 30 minutes after stimulation, you see very little, or not very little, but statistically significantly lower amounts of granule product in the cell. So when the neutrophils don't have ATG7, they don't secrete as much product in the supernatant. So we asked, what are the possibilities? Of course, you don't secrete if you don't synthesize the product. So the first possibility is that there is reduced synthesis. So the cell has decreased granular content. And the second possibility is that there is actually no defect in granular content. The cell has enough, but they just cannot secrete. There is an actual problem in secretion. It's very easy to test this. You just collect the whole cell and lyse the cells and do the same experiment. See what is the amount of the granules within the cell. I'm going to show you just one here, but we did this for all different types of granules. And you can see when you do the same gelatinous zymogram with lysate, now you can see the first three are the wild type. So this is the content with amount within the wild type neutrophils from 3 different mice. And these are 3 different knockout mice. And you can see they have much more, not less. And you can quantify this and see the intracellular content or intracellular gelatinous amount is actually increased compared to what was in the supernatant. So it's not that they don't synthesize, it's that there is a problem in retention. They cannot secrete. So the conclusion from all this was that ATG7 deficiency results in retention rather than a defect in synthesis. So no problem in synthesis, it's a problem of secretion. That's when we started to coin this secretory autophagy thing. But before going into autophagy, I told you that all these pathways, all these proteins have multiple functions. So how do we know this is autophagy pathway and not autophagy independent function of ATG7? So we decided to take another gene, ATG5. Also, it is involved in autophagy, but other functions as well. The point at that time was that 2 was more than enough. Otherwise, you can take many genes and it will just increase the number. So we take a ATG5, delete it, same thing. So you have ATG5 sufficient neutrophils, ATG5 deficient neutrophils. And we again did the same experiment to study degranulation. And here I'm showing all of this together. When you stimulate ATG5 deficient neutrophils, they don't secrete enough. They don't secrete enough MMP9 gelatin. So we did the gelatinous zymogram, lactoferrin, or beta-glucuronidase. So the conclusion was the same. Both ATG5 and ATG7 deficiency reduced neutrophil degranulation. I'm not showing here as well, but same thing for ATG5. There is no defect in synthesis. It's a defect with retention. They cannot secrete, release these granules within the supernatant. So the overall conclusion was that genetic ablation of the autophagy pathway reduced neutrophil degranulation. So now it became really interesting. So we have autophagy, which was considered an intracellular degradation pathway. And here we have this, that this pathway is somehow preventing secretion. So what's happening here? So far, genetic ablation reduced neutrophil degranulation. And this is a problem of degranulation or granule secretion. It's not a problem of synthesis or anything. So the first question we asked, is it a global defect or is it a specific defect? So what exactly I mean by global defect? It means that in the absence of autophagy, the neutrophils are largely non-functional. If that's the case, it's not very interesting from a paper point of view, still very interesting for scientific point of view. But if it turned out that the neutrophils are normal, but they have a very specific defect in secretion, then it would be much more interesting to find out how this is happening. So to do that, we did a very simple experiment. What we did, we take out these control or knockout neutrophils and added zymosan A. Zymosan A are just yeast particles. These particles in this experiment was coated with a green color called PHD. So now this is pathogen. So the neutrophils will immediately go and phagocytose this pathogen. But at the same time, the neutrophils, as I mentioned, the first thing they do when they encounter a pathogen, they secrete. So they will phagocytose and they will secrete their granules. So when we check the neutrophils, we'll be able to determine how much they phagocytose. And when we take the supernatant, we'll be able to determine how much they secrete. So in the same experiment, this would be the best control, would be able to figure out whether there is a global defect in phagocytosis and secretion or just a very specific secretory defect. So here I'm showing the results. It's a flow cytometry plot. In the x-axis, LY6G is the marker of neutrophils. So everything that's positive for LY6G are neutrophils, and PHC is what marked zymosan. Double positive means these are the neutrophils that have already phagocytosed those yeast particles. And this is the control, this is the knockout. And you can see when you look at the percentages, there is not much difference, actually slightly higher, though not significant. So there is no global defect in these neutrophils. They can clearly take up the particles. So the defect is in neutrophil degranulation is not part of a global dysfunction in these cells. It's a very specific defect in secretion. If that's the case, then if we assay the supernatant, then in the same experiment, in response to this zymosan particles, the neutrophils would not secrete enough. And that's exactly what we found. When we assay the supernatant for zymosan, you can see these four are wild-type neutrophils, so they have the autophagy pathway intact. These are the knockout neutrophils. Compared to the wild type, there's much less secretion by the knockout in the supernatant. So autophagy deficiency reduced neutrophil degranulation without affecting phagocytosis. That was one of the major findings of these studies. So if that's the case, the neutrophils are not secreting, so what? Neutrophils are the most important cells in the blood in the sense that they are like 60% of the blood cell. So what happens when the neutrophils are not good enough in secretion? What's the physiological significance? So we did many experiments to show that when you don't have neutrophils that can secrete, the inflammation, they cannot mediate enough inflammation. I'm just going to show two different experiments to compare this. The first one is a PMA induced ear inflammation. PMA is formyl myristate acetate, one of the strongest known inflammatory agent. Experiment is very simple. You add PMA on the ear. Within 4 to 6 hours, you see the whole ear becomes red and thick. It's like literally swollen up. And you just measure how much is the swelling in the ear. And you also do this imaging I'll show you that can directly visualize the inflammation within the ear. And when we do that, you can see the amount of ear swelling was again significantly lower in the autophagy or ATG7 deficient mice. So if the neutrophils don't have ATG7, they cannot mediate this ear swelling. And when you image this, you can see this is the amount of inflammatory product. In this case, this depends on oxidation. So the neutrophils secrete a lot of reactive oxygen species that oxidize the other granular product and give this bioluminescence. So in wild type, you see a lot of bioluminescence. In the knockout, very little. again showing the same thing, that the neutrophils are not secreting, not mediating inflammation. Remember, this depends on reactive oxygen species, because this becomes the theme of how autophagy is linked to this granulation or degranulation process. The other mouse model I want to point out, because this will be very important for the dendritic cells also. This is a mouse model of multiple sclerosis. It's called EAE. It's an autoimmune disease. So what we did, we had this conditional autophagy knockout mice where ATG7 is deleted from the entire myeloid compartment, macrophages, neutrophils. So you immunize with this MOG, myelin oligodendrocyte protein, and pertussis toxin. And within 7 to 10 days, the mice started to develop paralysis. Why that happens? This disease is based on two different components. The first one, this is true for all autoimmune diseases. But again, today's focus is on the innate immune side. The first part is called the initiator arm. The dendritic cells present antigen to activate CD4 or CD8 cells. In this case, this disease is mediated by CD4 cells. So the dendritic cells activate CD4 cells. CD4 cells migrate within the central nervous system. They break down the blood-brain barrier, go to the central nervous system, brain and spinal cord. And then these are CD4. So these are helper T cells. These are not cytotoxic killer T cells. So they cannot cause the disease by themselves. What they do, they recruit effector cells. And one of the most important effector cells are neutrophils. So once neutrophils enter into brain and spinal cord, they start massive degranulation. And that degranulation kills, demyelinate the entire central nervous system. And that results in paralysis. So again, initiator arm, effector arm. In the mouse model I'm showing right now, it's the defect in this effector arm, macrophages, neutrophils. The initiator arm is normal, which means the disease is normally initiated, but the prediction is that there would be defect in the effector arm. So when we check whether it's true, here on the left side, I'm showing you the black points are all from ATG7 sufficient mice. So they increase their clinical score. This clinical score depicts the severity of paralysis. Higher score means more and more paralysis. In contrast, the red one, the autophagy deficiency, they actually show much less severe disease in these mice. Same thing with ATG5 deficient mice as well. The black curve again, control mice, they have so you can see a strong, very large paralysis in these animals. The green one, autophagy deficiency in neutrophil macrophages, much lower severity. So autophagy deficiency in these myeloid cells reduced severity of EAE. We further did antibody studies and showed this is predominantly because of neutrophils. But the whole point is that in the absence of autophagy in neutrophils, these mice cannot mediate inflammatory and autoimmune diseases as the control animals. So how is this happening? Again, the same picture. Somehow, all the studies so far, all these things have been attributed to the fusion. So it's this autophagy pathway, it's just membrane after membrane, they're forming vesicles, they're fusing with different vesicles. And we thought somewhere in this fusion process, there is a defect. And you remember I showed that picture that this secretion of or degranulation depends on the production of reactive oxygen species. It turned out that production of reactive oxygen species actually depends on fusion of two different autophagosomal components. I'm not going into the greater details of the molecular mechanisms, how or exactly what happens. So we thought if the autophagy deficiency, the neutrophils would not be able to produce enough reactive oxygen species. In turn, there will be less fusion of granules with other membranes, and that would result in less secretion of this granule. So we directly measured how much reactive oxygen species these autophagy deficient neutrophils can make. And when we did these studies with just cytochrome C oxidation assay, the black line again shows autophagy sufficient neutrophils. And this is normal. So when you stimulate them, they produce a lot of reactive oxygen species, gradually comes down. Autophagy deficient neutrophils never produce that much. Eventually they go up, but never. The total amount of reactive oxygen species is much less in these mice. So now we have a model where the autophagy deficient neutrophils cannot produce reactive oxygen species. They cannot produce reactive oxygen species, meaning there is defect in fusion of these granules, and the defective fusion of granules eventually lead to defective release of granules. So the granules don't fuse with plasma membrane, they stay within the cells and cannot release their products in the cell, extracellular system. So to further test, we did a very simple experiment. We used a drug called DPI that scavenges reactive oxygen species. It's reduced the amount of reactive oxygen species. And we used this for just plain black 6 wild-type mouse. So we take wild-type neutrophils, add this drug, and then stimulate. And then you can see, compared to the wild-type control or vehicle-treated animals, when you treat with DPI, it reduces secretion of gelatinase. Not as much as autophagy deficiency, not completely zero, but it's significantly reduced. Same with lactoferrin, same with beta-glucuronidase. So inhibition of reactive oxygen species production reduced neutrophil degranulation. So overall, the model we proposed is that somehow, it's still not known exactly how, but somehow autophagy regulates this NADPH oxidase-mediated reactive oxygen species generation. And this reactive oxygen species generation regulates granule fusion with the membrane, and that in turn regulates secretion. So if you don't have autophagy, low reactive oxygen species, low or fewer granule fusion, and few or little secretion. And this is what we had the highlight of this paper that we published quite some time ago, that autophagy regulates neutrophil degranulation by mediating NADPH oxidase-mediated ROS production. So as I mentioned, this was just one of the mechanisms. That's why it cannot explain why there is still so much degranulation. So in this invited review, we actually proposed multiple mechanisms that happen here. We just focused on this one arm in that Cell report paper, that's reactive oxygen species. But there are multiple other potential pathways that can have effects. I'll just say one thing about that humoral component of immunity. It's not just degranulation or secretion of granules. There is something within the cell called inflammasome. It's a multi-protein huge complex, and it's involved in secretion of two very important cytokines, IL-1 beta, IL-18. And we and others now have shown that in the absence of autophagy, there is also problem there. So in fact, autophagy somehow interacts with inflammasomes and initiates or inhibits IL-1 beta and IL-18 secretion as well. But that's just the point. So now let's focus on the last part. So I have shown phagocytosis, degranulation, and secretion. But one of the major things that autophagy does, actually, this is the only thing that was known when we started this project, that autophagy is involved in antigen presentation by dendritic cells and other cortical thymic epithelial cells, other cells as well. So what exactly is antigen presentation? So dendritic cells or immature dendritic cells phagocytose antigens and present on the surface to activate CD4 or CD8 T cells. I have shown this model before. This is the initiation of multiple sclerosis or EAE in mouse. This is the exact same thing that happens for all known disease that's mediated by adaptive immune system. The first thing called the initiator arm is always the professional antigen presenting cells that presents antigen and activates T cells. Unless that step takes place, nothing else will happen. So it was thought that autophagy would be involved, or autophagy was involved, but nobody knew if that process would actually be able to activate T cells and actually cause any physiological problem. Will there be defect? If there is defects in autophagy, will there be a defect in autoimmune diseases? or whether in the physiological context there would be nothing. So before going into the detail, I'll just briefly overview. There are two antigen presentation pathways, exogenous and endogenous, for MHC class 2 and MHC class 1. Just three important things. MHC class 2 is for extracellular antigen phagocytosed by dendritic cells presented on class 2 to activate CD4 T cell. Class 2 presentation is for extracellular phagocytosed antigen presented to CD4. Class 1 is intracellular or endogenous antigen presented on class 1 to activate CD8 T cell. So we asked what happens when we have a defect in autophagy in dendritic cells, whether the class 2 will be affected, so the CD4 mediated diseases will not take place, or whether the CD8 mediated diseases will not take place. So we actually studied this in an in vivo in the context of this multiple sclerosis. So our hypothesis was very simple. If you don't have autophagy in antigen presenting cells, for class 2 mediated or CD4 mediated disease, EAE, this defect in antigen presentation means the CD4s will not be activated. So we hypothesized that this autophagy mediates autoimmunity, class 2 mediated autoimmunity, by regulating antigen presentation by antigen presenting cells. So now we are using an autophagy deficiency in dendritic cells, ATG7 knockout in dendritic cells. I've shown this picture before. So there is reduction in severity of the disease. The black one is the normal control mouse, sufficient. The blue one is the ATG7 defective or ATG7 deficient mice. But this deficiency now is only in dendritic cells. The previous deficiency that I mentioned was in macrophages and neutrophils. What is really striking, okay, the defect, they also have lower score, but now there is a defect in incidence of the disease. This is in complete contrast with what I have shown before. Before I have shown macrophages and neutrophils. There is a defect in the severity of the disease, but not the incidence of the disease. So what does this mean? So when I say incidence, it means if I have 10 mice, I immunize 10 mice, they all show the same disease. Everybody is diseased, but it's just lower severity. Here, if I have immunized 10 mice, that simply means 5 of those will have disease. The other 5 never even showed any signs of disease. So that's actually more explained in this figure. You can see the black one is a survival curve for the wild type mice. Every single animal, so this is a fraction disease free, which means at 0 means all animals now become disease with disease. So nobody is disease free anymore at 14, 15 days. So wild type animals, almost 100% of these always suffer from the disease. But when you have this ATG7 deficiency in the dendritic cells, quite a few will never show any sign of disease. This disease is EAE, multiple sclerosis, specific for only CD4 or initiated by CD4 T cells. So now we have two different things. Autophagy deficiency in the initiator arm will inhibit or reduce CD4 T cell activation. It will reduce incidence of the disease. And autophagy deficiency in the effector arm never has any problem in the initiation, but it only shows score reduction. Autophagy in neutrophil and macrophages mediates the effector function of the disease. So it's only for CD4 mediated disease, because when we did the same model in a completely different thing, sorry, not the same model, it's a completely different model. It's called Contacthypersensitivity. It's mediated by CD8 T cells. You immunize with one antigen and then challenge with the same antigen on a different site after 7 days. We had 2 different antigens, and then it's ER challenge. So again, measure ER swelling in a completely different, this is now antigen mediated challenge. There is no difference in ER swelling. So the overall conclusion is that class 2 mediated antigen presentation is at least modulated or regulated by autophagy. Class 1, not so much. So the diseases that are initiated by CD4 T cells have a large impact, autophagy has a large impact on those diseases. And then different pharma companies came and started to think about inhibiting autophagy to treat those kinds of diseases in different animal and mouse models. So to summarize everything that I have shown so far, in innate immune cells, autophagy first I have shown in epithelial cells. It regulates, for example, Pseudomonas infection. This is the example I showed, but it's not Pseudomonas alone. Multiple cells, almost all cells. If you don't have autophagy, you have more bacterial infection. If you have intact autophagy, somewhere in the pathway, they always control infection. And I have shown in macrophages, it's phagocytosis of MTB by a completely non-canonical mechanism. It's an unwanted or unappreciated effect of protein removal by autophagy. That's the original purpose. So autophagy removes degraded proteins, organelles. If you don't have autophagy, you have a large leftover of trash within the cells, and those trash things do other effects. One of those was phagocytosis of tuberculous bacteria, and also IL-1 beta and IL-18 secrete. And the other thing, in dendritic cells, autophagy regulates class 2 antigen presentation and activation of CD4 T cells. So the initiation of a lot of autoimmune diseases that are mediated by CD4 T cells were regulated by autophagy. And in contrast, autophagy in neutrophils, which regulates the secretion of neutrophils or degranulation, and that in turn mediates in neutrophil-mediated inflammation. Point to note, neutrophils are the first line of defense in our body. So whenever there is any microbes, anything in the cell, the neutrophils go there, secrete, and immediately start to attack the bacteria. So if you don't have autophagy in neutrophil, that first line of defense is severely weak. Neutrophils cannot mediate their function properly. So later on, as I mentioned, this whole pathway in neutrophils, other labs have shown the same thing in eosinophils and basophils and mast cells also as well. And this whole thing started as a completely separate pathway. It's no longer just autophagy, is no longer a stress recognition. It's a pathway called secretory autophagy. Somehow autophagy mediates secretion. Different cells, different mechanism, but the overall theme is the same, secretory autophagy. So this is the overall summary of the presentation, how autophagy mediates different innate immune functions in different innate immune cells. And this was the lab. This is the picture I could find. All this work was done in Baylor College of Medicine. This was my previous PI, Dr. Tony Isa, my thesis committee, and all the collaborators we worked on there. So with that, I'll just end here, and I'll be happy to take any question.
Thank you so much, Abhisek. It was really a very, very wonderful presentation. And the way you've explained everything, it was almost like a student attending a teacher's class. I'm extremely proud of you for making such a wonderful presentation, but I still would like to take up a few questions if they have any. So for doing this portion, may I request my colleague, Dr. Deep Chandan Chakraborty, to kindly do the needful. In case students have any question, it was very informative, but yes, very simple and lucid way that you've taken this presentation ahead. So it was kind of self-explanatory. It was wonderful to hear you out. Over to you, Deep Chandan. You can take up questions if students have any to ask.
Thank you, Sriparna Di, and thank you, Dr. Bhattacharya. It is a nice presentation. Oh, sorry.
It's okay.
Abhisek, it is a nice presentation, and it's actually take me back to the days of immunology research, and it is very nice to hear a very good basic science presentation when actually we are actually just waked from morning, and it is also for you to just delivering a very beautiful lecture before going to bed. So being a doctor from background, you actually started your life maybe from investigating from outside, and now you're investigating inside of the body. And it is a fascinating presentation. And I would say there are some questions from our students' end. One from my end, that is, you said that TLR is associated with autophagy, that you have shown TLR2, TLR maybe 4, I think, in some cases.
Most.
That means whether TLR can be in models where TLRs are negative or TLRs are deficient, these systems cannot respond through autophagy.
So that's a very interesting question. The question is about the contribution of TLR to the autophagy pathway. So TLR or TLRs, so our lab was the first one to show TLR4 that induces autophagy in response to LPS. Then Vojo Deretics lab did a complete study with all the TLRs, and it turned out of 11, almost all 11 TLRs could induce autophagy when they're stimulated appropriately. But TLRs are not the only thing that induce autophagy. As I mentioned, so there are many pattern recognition receptors, intracellular and extracellular. For example, TLRs are one of the major one, but inside the cells you have all those things, and they all will induce autophagy, because autophagy is one of the most evolutionary conserved mechanism. And all these mice I showed, these are conditional knockout, because if you delete autophagy from germline, so the whole mouse has no autophagy genes or no autophagy pathway, the mouse dies within the first 24 hours. So cells have evolved mechanism by which they multiple pathway or multiple stimulators will induce autophagy. So what we found, or what I found in my studies, if you don't have TLRs and stimulate the cells with TLR agonist, of course the cells will not induce autophagy. But if you induce the same cells with interferon gamma or other stimulator, there is no problem in autophagy induction. Or starve, what is the most important and most well-characterized, starvation. You just put the cells, no TLR, just put the cells in HPSS or PBS, 2 hours, they will induce autophagy.
Okay, thank you.
In the absence of TLRs, only the TLR inducing agent will not induce autophagy.
Okay. And one more thing, that is how viruses reacts to it. Some virus have ability to block the autophagy pathway.
Exactly. Yes. So this is a very, very interesting question, and completely different areas of research. Vaccination involves autophagy pathways these days. So how viruses, or what is the interaction between viruses and autophagy? A lot. So I will just say two different things for that. Host and pathogens co-evolve. So when you have an intracellular pathogen like a virus, most of the time the cells try to digest or clear that virus through autophagy pathway. So what the viruses started to do, they do two things. Some viruses evolve mechanism that will actively inhibit autophagy pathway.
Yes.
What is really interesting though, I showed so far all these degranulation. So if you have autophagy, the cells can release these granules. If you don't have autophagy, they don't release granules as much. Some viruses hijack this pathway. They get inserted, or they insert themselves into the granules. So when the cells have autophagy, they will release the granules, and the viruses will come out of the cells within the granules, infect other cells. So it's a very complicated host pathogen interaction. The way cells try to clear viruses try to adapt to the pathway, either inhibit or adapt to for their own proliferation and things like that.
Okay, thank you. And some from students end, one from Sriparna, maybe she has actually collected from her friends. That is said how the mechanism of initiation of autophagy different in the case of different biological stresses. I think you have actually told right now.
So this is again, so how autophagy is initiated is an enormously complicated process. What is most well characterized is mTOR, mammalian target of rapamycin, mTOR to inhibit mTOR2, ALK1. So multiple products are there. They all converge at some point to initiate the pathway. The overall structure of the pathway is the same. You have an isolation membrane that will, multiple proteins will come to that tiny little membrane and make it a really spherical circular autophagosome. The proteins that do this are the same, but how they are activated in the first place is completely different. So something that induces reactive oxygen species will induce autophagy by one protein. Hypoxia, which will lower reactive oxygen species, will induce autophagy by other mechanism. Damaged mitochondria need to be cleared from the cells. They will initiate autophagy by a completely different protein set, pink protein. So each of these set will initiate autophagy by completely different proteins, but the eventual result is the same. At some point, these pathways will converge and initiate the ATG5, ATG7 mediated system to initiate the whole path. Upstream signals are different, downstream pathway starting from yeast to humans exactly same. That's one of the most evolutionary conserved pathways.
Okay, thank you, Abhisek. And one question from is it DPI reduces the production of ROS? How the process, how it happens?
So DPI actually doesn't reduce the production. It's actually called a ROS scavenger. So instead of, when you have NADPH oxidase, it produces reactive oxygen species. DPIs just go there, bind to that and neutralize that reactive oxygen species. So immediately the oxygen species is formed, neutralized, formed, neutralized. So you never get enough reactive oxygen species within the cell.
Okay. Okay, and last one from me, that as you have told that ATG7 is sometimes it is a candidate which we often measure to show the amount of autophagy is happening. So can we expect something downstream which tells us that the way we can control autophagy in certain diseases, that the disease is going to be controlled? Is it a correlation coming in future?
It's actually there already, but it's not as simple. So why is that? So one of the ultimate thing that happens is that this autophagosome fuse with the lysosome and degrades the product.
Yes.
One of the most well characterized inhibitor of this pathway is something that we Indians have been using for literally hundreds of years. Chloroquine or quinine, anti-malarial drug. So it turned out chloroquine is not just an anti-malarial. You have seen like hydroxychloroquine in COVID. Yes. Chloroquine has also been actively used for multiple inflammatory and autoimmune disease, including lupus. Nobody knows how chloroquine controlled inflammation, but chloroquine is very well suited to control inflammation. It does multiple things, including inhibition of autophagy. The problem with specific autophagy inhibitors, though these are in clinical trials, is that autophagy is very important cell survival mechanism. So if you want to inhibit autophagy, the cell will die. Any stress and the cell will die. So it has to be a selective inhibition of autophagy. And to my knowledge, till date, those are just in preparation. So not any specific drug that specifically inhibit autophagy in one type of cells in one disease, but not in other. I have not shown it here, but chloroquine can actually reduce severity of EAE. I have done it, it's already published. So SLE, arthritis, collagen-induced and rheumatoid, in SLE I mentioned, and EAE, all three classical autoimmune diseases, when treated with chloroquine, reduce the severity of the disease. Maybe it's by autophagy, maybe by other mechanism. Nobody knows yet.
Yes. Thank you. Thank you once again. And now I would request Dr. Sajal Bhattacharya, our senior faculty. He would give you actually the vote of thanks. SB sir, are you ready, sir? Sir, are you there, SB sir?
Yes. Thank you very much, Dr. Chakraborty.
Sir, can I just intervene for a second? SB sir?
Yeah.
Okay.
Yeah.
So I'll just take a couple of minutes maybe to just introduce you, because Abhisek is kind of not aware of you right now. He has not been your student in the past. So Abhisek, thank you once again আচ্ছা, বিফোর স্টার্টস গোজ ahead with the concluding remarks and everything, এটা নতুন করে বলার কিছু নেই, আমাদের কাছে এসবি স্যার ইজ এ ভেরি নোন ফেস, ঠিক আছে? আমরা 2017 থেকে আশুতোষে আছি, বোথ মাইসেলফ অ্যান্ড দীপ চন্দন, আর এখানে তো অনেকে আছে যারা স্যারকে বহু বছর ধরে চেনে, ঠিক আছে? বাট জাস্ট ফর বিং দি আনভার্স, তোমার জন্য জাস্ট আমি শেয়ার করতে, জাস্ট আ ভেরি পার্সোনাল থট দো, মানে নাথিং প্রফেশনালি দের, এখানে বলতেই পারি আমি, ইট ইজ জাস্ট আ ভেরি, মানে বলবো হচ্ছে কাইন্ড অফ আ গডফাদার লাইক ফিগার টু আস in the department. Sir has been in the department for very long to be honest, and right now he is not only an associate professor in the Department of Zoology, but he also happens to be the convener of our postgraduate cell. So আমাদের Asutosh College we have 6 PG departments, and it's only under his guidance আর ওনার kind of বলতে পারো হচ্ছে whatever dreams he had way back in 2009 যখন থেকে Department of Zoology PG start হয়েছে আর কি. And not only the teaching part of it, it takes me immense pleasure to let you know, even at this age and time, sir is actively involved in research. ঠিক আছে, তুমি কথা বললেই বুঝতে পারবে স্যারের সঙ্গে। ইনফ্যাক্ট এই প্যান্ডেমিক এরার মধ্যে it's been more than a dozen papers that he has published only on the coronavirus, and few of them also have been kind of cited by WHO. So you can kind of extend the, imagine the kind of contribution that he has already. তো স্যার pardon me, আমি জানি আপনি এগুলো পছন্দ করবেন না, but still then আমার মনে হলো তো আপনাকে চেনেন না, তো just kind of sharing my thoughts here. So over to you. এবার স্যার আপনি যেভাবে
pleasure.
Absolutely, absolutely. It's a pleasure having him here.
Really honored, humbled, and at the same time, I'm a little bit embarrassed also for such a good and kind words.
No, sir, absolutely not. My pleasure.
It is indeed a great pleasure and proud privilege to be in this webinar, listening to an outstanding talk delivered by an extremely talented, brilliant scientist, Dr. Abhisek Bhattacharya of National Institute of Health, USA. Our HOD, Dr. Datta Ray, has already stated about his illustrious career, a perfect blend of medical science and biological science. He talked about the autophagy in the innate immune system. He stated elaborately the autophagy pathway, how innate immune cells work, and various aspects like sense danger, stranger aspect, adaptive immune response. Today, this is a very important area of immunological research. I thank Dr. Bhattacharya on behalf of all the stakeholders, professors, supporting staff, and students of the Department of Zoology, Asutosh College, for accepting our invitation and sharing his knowledge with us, which will help in furthering our understanding about the subject. I thank the college authorities for their encouragement, Vice Principal, Sir, IQSC coordinators, and for their encouragement and necessary support. I thank the technical support staff for their assistance. I don't want to encroach your time. I guess it's midnight, is approaching at your place. But while we are passing through a time which is so stressful and distressing due to COVID pandemic, your talk in this morning comes as a high octane dose of freshness and fragrance, scientific fragrance. So no more time. I would like to take a very good night to Dr. Bhattacharya, because I guess midnight is approaching to your place. Thank you very much.
Thank you. The pleasure is all mine. This has been an honor, truly.
Thank you so much, Abhisek. Thank you for taking out time for all of us, and looking forward to many more. তোমার সঙ্গে আগেও কথা হয়েছে, স্যারের সঙ্গেও কথা হয়েছে। ইনফ্যাক্ট তোমার শুনলে আরো ভালো লাগবে, দ্য স্টুডেন্টস যেগুলো কোয়েশ্চেন তুমি কাইন্ড অফ, যাই হোক যতটুকু ডিসকাশনে আমরা আনতে পারলাম, মোস্টলি দে ওয়ার ফ্রম দ্য পিজি স্টুডেন্টস, ঠিক আছে? তো আমি এই টাইম বিং এর মধ্যে আমি আন্ডার গ্রাজুয়েট স্টুডেন্টদের সঙ্গে, দে আর ইন দ্য ইউটিউব লাইভ স্ট্রিমিং রাইট নাও, তো আমি ওদের সঙ্গে ইন্টারঅ্যাক্ট করলাম, এন্ড ইট ইজ ওয়ান্ডারফুল টু শেয়ার ওদের বক্তব্য। ম্যাম, স্যার এক্সপ্লেইনড ইট সো ওয়েল, ওই জন্য আমাদের এই মুহূর্তে কোনো কোয়েশ্চেন নেই। I do realize they have to study a lot more to get to the level that you're doing right now. কিন্তু আমার শুনে ভালো লেগেছে that they could understand the talk and they could even take home lesson গুলো।
In future we can collaborate with him.
Absolutely, absolutely. I'm looking forward to some kind of theoretical sessions আমি বলবো আর কি, not only through the webinar but active classes এর মধ্যে। তো সেটা আমি কথা বলবো, especially for our PG department তো নিশ্চয়ই।
That would be a great pleasure
as well as in neuroscience. It will be an honor for us as well, Arki.
Yeah.
So thank you so much. Please take rest, and পরে we'll catch up again. And thank you everybody. সবাইকে আজকে এখানে জয়েন করার জন্য
Thank you everybody.
Thank you
And
so much.
anytime,
Thank
Dr.
you.
Didi, I'll be happy to help in any way I can.
Thank you so much. Thank you so much. Thank you. I'm looking forward to many more of these. Thank you so much. Thank you. Thank you and good night. Good night.
Good night.
Thank you. Thank you. Good night.
Thank you everybody and faculty members যারা আজকে আমাদের সঙ্গে আছেন, and especially the students both UG and PG যারা আমাদের সঙ্গে এখানে জিমিতে আছো বা ইউটিউবে, thank you all for joining, and especially technical support যেটা স্যার বললেন, সুমনের নাম তো নেব, আর কৌস্তব আমাদের সঙ্গে থ্রুআউটই আজকে আছে আর কি। So thank you so much for taking the labor and pain for us. Thank you.